Hello, dear students. In this lesson, we will focus on the evidence supporting the theory of evolution. At the end of this lesson, you will be able to explain how fossils, anatomical and molecular records, and other pieces of evidence support evolution. This is this lesson's essential question. What are the different pieces of evidence supporting the theory of evolution? Why is evolution such a well-known idea in biology? Most people have heard that modern organisms evolved from earlier, extinct ancestors. However, the familiarity of evolution does not always mean understanding. Many people see evolution as controversial, not because of the science, but because they think it conflicts with personal or religious beliefs. Biology does not ask what we believe. It asks what the evidence shows. This list summarizes the major categories of evidence for evolution. Scientists do not rely on just one source. Instead, they examine fossils, body structures, molecular data like DNA, and even changes caused by humans. Each type of evidence comes from a different field, yet they all support the same conclusion. Populations change over time. That agreement across fields is what makes evolution such a strong scientific theory. The fossil record gives us the most direct evidence for evolution. Fossils are preserved remains or traces of organisms that lived in the past. Today, scientists have access to far more fossil discoveries than Darwin did, allowing clearer evolutionary patterns to be seen. The whale fossil shown here is especially important because it contains pelvic bones and hind limbs, linking whales to land mammals. This helps explain how major evolutionary changes can happen gradually over time. The images show that fossils come in several forms, not just bones. Some fossils form when minerals replace organic material, like petrified wood. Others preserve traces, such as footprints or burrows, which show how organisms behaved. Some organisms are trapped in amber, ice, or frozen soil, preserving incredible detail. Together, these fossil types help scientists reconstruct ancient environments and evolutionary history, not just what organisms looked like, but how they lived. Paleontologists gather evidence from fossils in the field and laboratory. Careful excavation allows scientists to preserve the position and condition of fossils. The image of a dinosaur fossil with eggs suggests nesting behavior and parental care. From bone placement and surrounding evidence, scientists can infer how an organism lived, moved, or even protected its young. Fossils are not just bones. They are clues to past behavior. This slide highlights transitional fossils, sometimes called missing links. These fossils show intermediate traits between earlier and later species. The whale fossils shown here reveal a gradual shift from land-dwelling mammals to fully aquatic whales. Early forms had legs and could walk, while later forms were better adapted for swimming. These fossils provide a clear sequence of change, showing evolution as a gradual process rather than a sudden event. This slide presents one of the most complete fossil records, the evolution of horses. Modern horses belong to the genus Equus and descended from much smaller ancestors. Over about 55 million years, fossils show changes in body size, teeth, and limbs. These adaptations reflect shifts from forest environments to open grasslands. The fossil record, although powerful, is incomplete. Fossilization is rare because soft tissues usually decay before they can be preserved. In addition, movements of Earth's crust can destroy fossils or bury them deep underground. Fossils also form only in certain environments, such as riverbeds or shallow seas. Finally, scientists have not excavated every location on Earth, so the absence of a fossil does not mean an organism never existed. The diagram on the left explains how fossils are formed and how scientists determine their age. When an organism dies, it may be buried by sediment, which protects the remains. Over time, layers of sediment build up, and the remains fossilize. The diagram on the right shows fossil succession, meaning older fossils are found in deeper rock layers, while younger fossils are found above them. This slide introduces transitional fossils using Australopithecus afarensis as an example. Transitional fossils show traits shared by both earlier ancestors and later descendants. This species lived millions of years ago and had a mix of ape-like and human-like features. It could walk upright, but still had features adapted for climbing. This shows Archaeopteryx and places it in historical context. It was discovered in 1861 and immediately supported evolutionary predictions. Scientists had proposed that birds evolved from dinosaurs, and this fossil matched that expectation. Since then, many additional transitional fossils have been found. This slide presents Archaeopteryx. Its skeleton shows wings and feathers, which are bird characteristics, but it also has teeth, claws, and a long bony tail, which are reptile-like traits. This combination tells scientists that birds did not appear suddenly. Instead, they evolved from dinosaur ancestors. Archaeopteryx bridges the gap between two major groups, dinosaurs and birds. Tiktaalik is an extinct organism that lived about 375 million years ago. It had fish-like features such as scales and fins, but it also had traits seen in early land animals. These include a flat head, a neck, and strong fins that could support weight. Tiktaalik represents an important evolutionary transition from aquatic life to life on land. The diagram on the right shows a sequence of fossils illustrating the evolution of tetrapods. Early fish had fins suited for swimming, while later forms developed stronger limb bones, wrists, and necks. These changes allowed movement in shallow water and eventually on land. Each organism in the sequence was well adapted to its environment. Let us focus on anatomical evidence for evolution. As vertebrates evolved, the same body structures were adapted for different uses. Even when the function changed, the original structure remained recognizable. These are called homologous structures, meaning they came from the same body part in a common ancestor. This slide shows the forelimbs of different mammals, including humans, cats, whales, and bats. At first glance, these limbs look very different because they are used for grasping, walking, swimming, or flying. However, when you look closely, the same bones appear in the same order. These are called homologous structures, meaning structures that come from the same ancestral body part. Different functions can evolve from the same basic design. Let us compare skeletons of humans and other apes. Although they differ in posture, limb length, and movement, the same bones are present in both skeletons. The skull, ribcage, spine, arms, and legs all follow the same basic plan. These similarities indicate common ancestry. The differences reflect adaptation to different lifestyles, such as upright walking versus knuckle walking. This slide compares the skeletons and skulls of different primates, including humans and gorillas. Even though these species differ in size, posture, and movement, they share the same basic skeletal plan. The arms, legs, spine, ribcage, and skull are made of the same bones arranged in similar ways. These similarities are examples of homologous structures, meaning structures inherited from a common ancestor. Vestigial structures are part of the anatomical record. Many organisms possess structures that no longer perform their original function but resemble those found in ancestors. These structures remain because evolution modifies existing traits rather than removing them entirely. Examples of vestigial structures shown here include goosebumps, reduced eye structures in some animals, the human appendix, and muscles connected to ear movement. These structures resemble features that were useful in ancestors, but are no longer essential today. Their presence makes sense only through evolution. Evolution modifies existing structures rather than removing them completely. More examples include reduced ear muscles in humans and remnants of hind limb bones in snakes. These structures resemble features that were functional in ancestral species. Some whales still possess small pelvic bones that no longer support hind limbs. These bones resemble those of land mammals but have no clear function in swimming. Their presence suggests that whales evolved from land-dwelling ancestors. Evolution reshapes structures as environments change, but traces of earlier forms often remain. Embryological development is another evidence for evolution. During development, embryos often show traits that reflect ancestral forms. These traits may later be modified or lost as development continues. This pattern suggests that evolution works by adding new instructions onto older developmental processes. This slide shows embryos of different vertebrates at early stages of development. Even though these organisms become very different as adults, their embryos share features like pharyngeal arches and a tail. These structures appear early and later develop into different body parts. Such similarities are best explained by descent from a common ancestor. This slide shows embryos of different vertebrates at early stages of development. Even though these organisms look very different as adults, their embryos share similar features, such as pharyngeal arches and a postanal tail. These structures appear early and later develop into different body parts or disappear. These shared embryonic traits reflect common ancestry. Because organisms evolved from simpler ancestors, traces of that history are stored in DNA. When one ancestral species gives rise to multiple descendants, those descendants initially have very similar DNA. This high similarity reflects their recent shared origin. DNA acts like a biological record of evolutionary change. After species split from a common ancestor, they evolve independently. As this happens, mutations accumulate and DNA differences increase over time. Even so, shared sequences remain as evidence of common ancestry. Both similarities and differences in DNA help scientists reconstruct evolutionary relationships. Guinea pigs were once grouped with rodents like mice because they look similar and share some traits. However, when scientists studied their DNA and molecular structures, they found important differences. Based on this molecular evidence, guinea pigs were reclassified into their own taxonomic group. Modern classification relies more on genetic data than appearance alone. Although this slide shows a living horseshoe crab and a fossil trilobite, the strongest evidence linking them comes from molecular biology. Scientists compared DNA sequences and conserved proteins, meaning proteins that change very slowly over time. Horseshoe crabs share more similar gene sequences with spiders and trilobites than with true crabs, even though they look crab-like. This tells us they share a more recent common ancestor with arachnids. Humans share approximately 98% of their genes with chimpanzees and about 93% with macaques. These numbers reflect how recently each species shared a common ancestor. Because genes are inherited, closely related species retain more similar DNA sequences. Over time, mutations increase genetic differences. Scientists examine specific gene sequences and count how many nucleotides differ between species. Fewer differences indicate a closer relationship, while more differences suggest a longer time since divergence. The chart shows that humans are most genetically similar to chimpanzees, followed by other primates. This method allows scientists to build evolutionary trees based on measurable data. All living great apes except humans have 24 chromosomes. Humans have 23, not because one was lost, but because two ancestral chromosomes fused together into a single chromosome. Scientists can see clear molecular signs of this fusion, making it strong evidence of shared ancestry. Since chromosomes carry genes, changes in chromosome structure record evolutionary history. Artificial selection happens when humans intentionally choose plants or animals with desirable traits to reproduce. Over generations, those favored traits become more common in the population. This process is similar to natural selection, but the selecting agent is human choice rather than the environment. Here, we see a diagram illustrating artificial selection for plant height. In the original population, there is natural variation in height. Humans select the tallest individuals as parents, shown in the highlighted group. These plants pass on genes associated with greater height. In the next generation, more plants are tall, and the overall population average shifts upward. This demonstrates evolution at the population level, not within a single individual. Here is a powerful example of artificial selection using wild mustard. Instead of selecting the same trait, humans focused on different plant parts. Selection for leaves produced kale, selection for stems produced kohlrabi, selection for flower buds produced broccoli, and selection for apical buds produced cabbage. Although these vegetables look very different, they share the same ancestral species. This demonstrates how flexible genetic variation can be when guided by selection. Teosinte, the ancestor of modern corn, had small, hard seeds and looked very different from today's crop. By consistently choosing plants with favorable traits such as larger kernels and easier harvesting, humans altered the plant's genetic makeup over generations. This process produced intermediate forms and eventually modern corn. He slide also shows similar before and after changes in other foods. These examples highlight that evolution does not require millions of years when selection pressure is strong. All modern dog breeds share a common ancestor, the wolf, yet they show enormous variation in body size, shape, and behavior. These differences arose because humans selectively bred dogs for specific traits such as speed, strength, or companionship. On the right, sheep domestication shows a similar pattern, where selection favored traits like wool production and calm behavior. Even though these animals look very different from their ancestors, the changes accumulated gradually through controlled breeding. In cattle, different breeds were developed for specific advantages, such as tolerance to heat or quality of beef. By crossing breeds, humans produced cattle that carry multiple desirable traits. In pigeons, selective breeding focused on features like feather shape and body form, creating many distinct varieties from the same ancestral population. Scientists selected foxes based only on tameness, choosing the calmest individuals to breed each generation. Over time, the foxes became friendlier, but they also developed physical traits commonly seen in domesticated animals. This happens because genes often influence multiple traits at once. Selecting for behavior can therefore reshape both appearance and physiology. In experiments, farming, and domestication, humans repeatedly choose organisms with specific traits such as size, behavior, or productivity. Over generations, these traits become more common, leading to large and sometimes rapid changes in populations. This process works because traits are genetically inherited. The success of artificial selection demonstrates that selection is not just a theory, but a tested evolutionary mechanism. To answer the essential question, evidence for evolution includes fossils, biogeography, homologous structures, vestigial structures, embryological development, and molecular biology. To conclude, name and give example of one type of evidence for evolution, explain how it supports the idea of common ancestry, and explain which evidence you find most convincing. Thank you for listening.